So today I'm going to tell you work that I've been doing recently, and also, let's see, get rid of 1 second. Got my pointer on, which then takes control over the whole computer. So work that I've been doing that actually Juan and the people from his group were also part of. So it's fun to bring this story to you today. As you, any of you who've talked to me know that I tend to talk pretty fast, and I'm obviously speaking in English, not Spanish. So if anybody has questions along the way, feel free to interrupt, and I'm happy to either explain a point or restate what I say. So feel free to interrupt me. As you heard, I started really as a botanist, and I've come more recently into appreciating lots of other taxa, including termites. So I'll talk to you a bunch about plants overall and also termites today. And so, I mean, before I jump in, I want to start by giving you a little bit of a description of my lab group. And so I met a few of you in January when I was visiting, and I'm really pleased to meet more of you and unpack a little bit more my group. And so I've spent a lot of time. First, I was trained as a botanist, as I said, and was really thinking about the functional ecology and macroevolution of plants. And then more recently, I've added in trying to think about how fungi and bacteria, their functional ecology and macroevolution in termites. And toward these ends, I've done things like building big databases of plant construction, wood density, things like that I can explore across the globe. A lot of the then more field-based work that my lab group does, and lab-based group, is thinking about how these different organisms interact. So really thinking about how plant construction affects who it interacts with, some of these different decay agents such as termites and fungi, and what are the consequences of that plant construction on how fast and in what forms carbon cycles out of wood. And so I'm going to tell you a bit about a project that we call the Wood Termites and Fungi Project as we move along, as well as a global experiment that Juan and others were involved in right there. Okay, and so as you're thinking about my lab group, what we try to do is think about what leads to how plants are constructed, and then what leads to how they're breaking down. And so some of our early days, we did a lot to link up how climate affects how plants are constructed, the plant traits. And then that's led to them thinking about how direct effects of climate and direct effects of plant traits and affect how trees die, their mortality, as well as how they decay. So there's a lot about the death and destruction of the plant world that we think about in the lab group. Now, these are mediated by different mortality and decay agents that lead to the mortality and the decomposition. And these are shaped again by the climatic circumstances. I'll describe those today. And that'll affect how carbon is lost. And ultimately, there are potential feedbacks to the Earth system across this process. So in different projects, we've taken different aspects of this flow diagram to query more about plant construction and plant destruction. I'm seeing, let's see, there's 2 chat messages, making sure, okay, so that's all of you just hopping in. So feel free, I'll try to pay attention to chats too as I'm moving through. And so with a lot of my more recent projects, I've been using different ways to measure carbon flux. So here, this is an infrared gas analyzer that connects up to a chamber that measures CO2 or CO2 and methane flux from the soils, or using small sensors to measure ecosystem scale carbon fluxes. And so I've got a number of projects that I'm building upon, and these include a lot of these, probably some of the common themes are thinking about how carbon cycling is happening at biome interfaces where you have different systems meeting, and those biomes might be shifting spatially or temporally, and especially thinking about those systems in the face of global change. And so I'm here in Brazil building up a system in the Cerrado, which we have a flooding gradient. So you basically go from peatland to savanna that's mostly dry except for some through-fall rainfall. And but then you get seasonal shifts in rainfall. And so we're thinking about how wetting and drying affects the carbon cycling and how you shift between CO2 and methane flux. The system is also really interesting because there's these large earth mounds that are thought to be because of these really big termites that are on the landscape. And what these will then do is lift the earth above the flooded landscape and potentially cause shifts in carbon dynamics, but also the vegetation that's associated with them. When I was visiting you all in Colombia, I was lucky to go into the field with both Juan and Adriana. And so we've been talking about potentially starting some projects thinking about carbon cycling in the Paramo and the upper Andean forest. And then I've been thinking of systems that are shaped by tidal systems and sea level rise. So things like mangroves, salt marshes, and the fjords in Chile, as well as in Antarctica, where you have warming potentially causing shifts in the rates and forms of carbon cycling in these different systems. So you can see that in all of these, there's vegetation, there's decomposition, there's meeting of different systems, either spatial or temporal shifts. And a lot of this is this focus in the Latin America has been in the past year and a half. So I moved to University of Miami about a year and a half ago as the Aresty Chair in Tropical Ecology, which gave me a nice mission that I'm focusing in the tropics. I am calling the tropics pretty broad, as you can see, thinking about Latin America more broadly, but also places. I've worked extensively in Australia, and I've worked some in Africa too. So I think this has given me a nice research focus, as I was outlining for you. I also am required to teach an off-campus field course that I hope to start by developing in Brazil, but then potentially in other locations. And then the third part, which I talked a bit about when I was visiting folks there in January, is that the department has long wanted a tropical biology center. And I've thought hard about what I would like that to be if I were to start such a center. And for me, a lot of that is building bilateral exchanges with different project partners or different universities with pretty much friends that I like working with, that I think where we can benefit students at both of our departments by coming up with ways for training and courses, but also research focuses that let us think about tropical biodiversity in the face of global change. And so if any of these are of interest, feel free to reach out to me after my talk or ask me questions at the end of my talk today. So these are some of my missions for why I'm on sabbatical here in Brazil, but also have been traveling around Latin America and also South Africa, visiting different groups. And today I'm going to tell you a bit more about my research. And this is going to begin by giving you a background of decomposition. And so for some of you, this might be pretty remedial. And so hopefully, though, for some of you, this is all new information, thinking about how decomposition happens, and hopefully making you think about being as excited about decomposition as I am. I think it's one of those few places where you can really relate how plants are made for life to what happens in the carbon budget and their cycling and their afterlife. So then I'm going to tell you a bit about the history of the story itself, how it developed. Then I'll tell you about the recent paper that we had published, some about the authors, and finally some about this amazing illustrator that is going to be a bunch of these images that I'm going to show you across today. And so let's begin broad scale. So as I said, I was trained as a botanist, thinking about how plants are constructed for life. That's meant that I've gone and looked at micro to mesoscales to think about, why is wood built like it is? Why does it have the anatomy that it does, or the chemistry that it does to live in different parts around the world? But then I've become especially interested in thinking about what are the afterlife effects. So why does, after different decay agents come through, in this case fire, why do you have some pieces of wood still in the landscape and others that have burned away? What makes them more or less susceptible to things like fire? And so why does this matter? Well, forests are the largest above ground biotic store of carbon. There's about as much carbon in forests as about half of the CO2 that's up in the atmosphere. So there's a massive amounts of carbon in forests. This is true while they're living. So here's my field site in far north Queensland, Australia. This is Mount Sorrow. When Captain Cook was floating around looking at places, that he was quite sad, I think, or desperate when he was in Australia and New Zealand. And so we have Mount Sorrow that looks out across our field site in the tropics in Australia. So this is certainly true when you have living forests, but it's true once that wood senesces and the parts of the tree or the tree dies. So you can think about, maybe you have a dining room table that you inherited from your great grandmother. And so that table has persisted across decades. And so wood can store carbon for years to millennia as dead wood. And what really matters is how that wood is interacting with different decay agents to release that wood. So who are these decayers? Well, globally, microbes, fungi, but also bacteria are really important decay agents. You find them everywhere you look in the world. There are microbes present. In certain parts of the world, macroinvertebrates, insects start becoming important. Termites are probably the one that's most important of this group. So in the tropical regions, you can get termites that can really decimate wood and wood products. And for abiotic agents, you have fire. So in places where there's enough moisture to build up vegetation, but then you have a dry season that causes senescence, you'll get fuel loads being built up and fire can become important. And then there's other abiotic decayers such as solar radiation, leaching, freeze-thaw events and things like that can also cause decomposition. So all of these different decay agents are shaped or controlled in terms of their rates of decay by how the wood itself is constructed. We know the most about microbial decay. So microbes tend to really struggle with breaking down lignin. And so as you have lower total carbon and lower lignin concentrations and higher nutrients, decay rates are faster. Typically denser wood decays more slowly too. So we know something about microbial decay. We know less about how these other agents are affected by wood construction. And then environment controls both how that wood is constructed, but also which decay agents can be present. So precipitation is typically pretty important. We'll talk more about that as we move forward, but also temperature. And so one of the things that I'll talk to you today is about Q10s. So Q10 is that for a 10-degree increase in temperature, how fast does something happen? And so this is really related to enzyme kinetics. So for a 10-degree increase, enzymes typically have a Q10 of 2. They double the rates of that enzyme is working for a 10-degree increase in temperature. But we also see that processes such as decomposition have been described as Q10s or effective Q10s. And so I'll describe that in the talk today. So I'm going to focus in really on the biotic decayers. And for today, I'm going to focus on the microbes, the wood-dwelling microbes, as well as the wood-feeding termites that are decomposing wood. And I think that these are really important and interesting decayers, in part because they both go after the same substrate, but they're also really different in a number of ways that they handle and interact with the wood. So one of them is how they find the wood. And so microbes are, as I said, everywhere. They can fall down as spores and land on the wood. They can colonize from the soil as hyphae. There's also a lot of incipient microbes that are sitting in the wood as the tree is living and come out once that wood senesces. So you can't predict that any particular microbe is actually going to do the decomposition, but you can say that I can guarantee that there's going to be microbes in the system. Termites, on the other hand, these are searching animals. They're insects, they're ectotherms. What happens is you'll have a worker looking for wood, and then what it has to do is return back and recruit its friends, recruit other termites from the mound to then come eat the wood. So there's a two-step process. The first part is the termites have to discover the wood, and then the second part is when they actually consume the wood, either in the wood itself or bringing it back to their mounds. So they're different in their discovery. They're also different in how they release that carbon from wood. So microbes don't have stomachs. What they do is they excrete extracellular enzymes out into the world, and these enzymes then break down various carbon polymers using hydrolases and peroxidases and things like that that target particular compounds, and then they'll reabsorb, or they'll absorb up those products that they've broken down. Termites do have stomachs. In their stomachs, they have some of their own endogenous cellulases, but they also have, they keep a whole suite of different microbes that are able to help them break down wood. And so they're different in terms of the how they release them. They're also different in terms of how sensitive they are to different environmental gradients. So microbial decomposition is tied to precipitation. To get those enzymes to the substrate, you need moisture to move the enzymes around. Termites, on the other hand, they have moisture in their stomachs, so they're less sensitive to having the wood in contact with moisture. They also have various adaptations to handle lower moisture. So they'll put mud or sheeting across the landscape. They'll have runways where they protect themselves, and they typically live in mounds so that are less sensitive to changes in moisture. So there's differences in moisture sensitivity. There are also differences in temperature sensitivity. And so It's typically been found that for microbial decomposition, that at local to regional scales, you'll see that microbial decay has a Q10 of 2, which makes sense because these enzymes, the microbial decomposition is driven by these extracellular enzymes. So Q10s of 2 are typical for enzymes. Termites have not been studied actually until we were looking at them in terms of their Q10s, but we know that termites are largely relegated. You mostly find them in the tropics. That's where their diversity and abundance is greatest. So it seems like they should be pretty sensitive to temperature. And then they differ in terms of how they're actually unlocking the carbon from the wood. So carbon can be tied up in really hard things like lignin, as well as cellulose, hemicellulose, and labile sugars. Only some fungi, the white rot fungi, are able to break down lignin. So if lignin isn't broken down, it gets buried in the soil, it gets locked into the soil. But they'll, as they're decomposing carbon, they're going to be respiring and releasing CO2. So you can have some of that carbon going up as CO2, or depending on which microbes, you might get the lignin being broken down or not. Termites also release CO2. Their termites are unable to break down lignin, but the termites in Africa and Asia, some of them, especially Macrotermes, is a fungus farmer, and it farms a white rot fungus. So they'll bring the wood back to the mound, feed it to their garden of fungus, and then they'll eat that fungus. And so they're able to actually break down the lignin. The other thing that's interesting is termites are like little cows. They release methane. So they're essentially like, little farting cows that are on the landscape where they're releasing methane. Methane is a 30-fold worse greenhouse gas than CO2. So depending on who's touching the wood, is it microbial or termite, you can have really different rates and pathways that carbon's leaving the wood. So that's my background about decomposition. Does anybody have any questions about any of that before I hop on further? All right, well, feel free to ask as we go along. So there's your 101 intro course on decomposition. So let's move into the history of the story. And so as I said, I was really trained as a botanist. Juan and I were in grad school together, and back then I was actually in zoology, but I would spend all of my time in his lab in botany and studied trees there. And so trained as a botanist, I was building these big trait databases. And I started chatting with folks with Will Cornwell and Hans Cornelissen, who ran a working group where we wrote an empirical paper and a review paper where we thought about how plant construction can affect the rates of decomposition by different decomposers. That led me to set up a pilot study with my first job my first faculty job in St. Louis, Missouri. St. Louis, Missouri, it's in the temperate zone, so microbes are really the drivers there. So I was associating microbial decay with plant construction. Just after that, I started working with Jeff Powell as well as Will Cornwell in Sydney, where Sydney is temperate zone, but you start getting more termites, and then fire is also really important. And so we were relating plant traits to decay, but as we started adding in other decayers. And then most recently, I have an NSF NERC grant where with Jeff and Will, as well as Paul Eggleton and Steve Allison, we're working in far north Queensland, and we're in the tropics there. So lots of termites can be found. And so there we're looking at the relative roles of wood feeding termites and wood dwelling microbes in terms of the decomposition, working along a rainfall gradient. So we've had these series of projects where I've asked some questions, learned something, and expanded out to new systems. And so with this project, what our expectation is the termite role is going to be relatively more important and in dry seasons and in dry sites because of their adaptations they have for low moisture. So in this project, we've been looking at the rates that carbon turns over, the fates of that carbon. Does it leave as CO2 or methane, or does it get locked into the soil? And then we're working on an ecosystem level model, taking all of our empirical data to try to understand carbon cycling from wood at the ecosystem level under current and future climates. So this has been our goal. This is our gradient. So we're up here in far north Queensland. We have a couple of savanna sites, a drier and a wetter savanna site, an in-between sclerophyll site, and then a drier and a wetter rainforest site. So we have about a 10-fold change in rainfall across our gradient. As we were trying to get funding from NSF and Australia Research Council, ARC and NERC in different places, we kept getting told that termites didn't matter that much. So why are we focusing on them? So we decided we had to do a pilot study to kick this off so that we could demonstrate that we should be getting more funding to look at the relative roles. And so with this, we did a pilot study and we published a method which we called a woodblock test. And this was in collaboration with Alex Cheeseman and Lucas Cernasak at James Cook University. In this project, what we did was we took radiata pine because it was novel at all of our locations, and we wrapped it in mesh that kept out the termites, and then we cut holes in half the bag so that termites could get in. And so we were able to demonstrate variation across seasons and to some degree across sites at the endpoints of this gradient that I just showed you at our wettest and driest site. And that then ultimately led to us getting funded by NSF and NERC, the UK funding agency, to ask all sorts of questions about the amount of carbon that's being stored in wood along our gradient and the different roles of microbes and termites. And so we did things like we had to, across all 5 sites, we wanted to know the amount of living and dead biomass that was there. So we set up plots to look at the living and dead biomass. We found that there's greater living biomass and there's also dead biomass in the rainforest and the savanna sites. But that it was interesting, the ratio of living to dead material was the same. We also knew from some of our earlier work that inside trees, you get various heart rot, hollowing of trees. You also have in other places termites that eat the inside of trees. This has been for me, one of the most interesting findings of our study. So I'm going to tell you more about it in a minute. So we were looking at the internal stem damage of trees. I'll continue that story in a minute. We also put out pine blocks across all 5 sites, as well as looking at the native stems that we harvested and put out to look at rates and forms of decomposition and how termites and microbes were differentially affecting the decomposition. So we just had a paper come out in the past couple of weeks with one of these studies where we were looking at the native stems. And what we found was that we could predict microbial decomposition from our wood traits, the chemistry, the chemistry of the wood, things like the syringeal to guaiacal lignin concentration or the amounts of nutrients. We could predict the microbial decay really well, but we weren't able to predict termite-driven decay very well. In part, we had pretty low termite hits, although we had relatively high termite discovery. So I have some thoughts about this and happy to chat more about that with you. My former graduate student, Becca Clement, spent her dissertation looking at the termite fauna across this gradient. And what she found was that there was a huge turnover of the termite species, but also the guilds as you moved across from rainforest to savanna. So you had different species, but also different types of termites that were really important across these landscapes. And then as we harvested each of the pieces of wood, we would measure the CO2 and methane efflux off of them, as well as measure things like the enzyme expression, the microbial community composition, and the changes in chemistry. So we found interesting things that every time we had a methane spike, we found termites in that piece of wood, or if we found termites, we were going to find methane. So it's generally thought that soil and litter feeding termites produce more methane than wood feeders, but we were definitely getting methane efflux from the soil feeders. One of the things, as we were setting up the project, we were realizing was that termites, unlike microbes that stay their whole lives in the wood as they're doing decomposition, the termites often will take bites and bring that wood back to their mounds, and then the decomposition happens often inside the mounds. So we set up some mesocosm experiments where we grew termites in buckets and we fed them different species of wood to see how fast CO2 and methane would flux. And we found some really interesting differences in terms of the total gas fluxes, as well as the relative concentrations of methane to CO2, depending on which species of wood we are feeding them. So let me do a quick deeper dive into the internal stem damage, because for me, I think this has been the thing that I expected least and have been most fascinated by. And it's really my graduate student, Abby Yatsko, is following up on a lot of this. And so as I was describing from earlier work where we were taking increment cores, we knew that we would find, especially in the rainforest, certain trees would have lots of heart rot in them, different fungal damage. We'd have liquid pouring out of these that smelled pretty disgusting. And then we knew from especially the savanna sites that we had this termite, Coptotermes acinasiformes, that liked to hollow the inside of trees, build mounds at the base and then hollow them. Here's where we took a core, and then by the next day, they'd already mudded over this hole that we created. This is so common that the traditional landholders in Australia make didgeridoos. So didgeridoos are termite-made musical instruments because they know that they can cut down these trees and find hollowing that leads to interesting musical sounds. So what we did was across our gradient for about 300, I think 300 trees, 300 or 400 trees, we used a resistograph drill that has a small stylus that gives you resistance across the stem as you move from the bark to the inside of the tree. And we related this internal stem damage to various site and stem level predictors. And so we thought that at the site level, precipitation could be really important because we know that we get this turnover and the relative importance, or what we were seeing is heart rot by fungi versus the piping of termites inside the tree along our gradient. The resistograph drill doesn't tell us who does the decomposition. We can just look at how much resistance it has as it moves across the trace. We thought that one of the ways we could think about is that if termites have high amounts of damage in the down deadwood, they're likely going to have high amounts of damage in the standing trees. So we used Becca's measure of the amount of termite damage in down deadwood at each of our sites from surveys she did of the termites. So those are our site level predictors. And then we had stem level predictors. We thought that maybe bigger trees, since they're either living longer or have greater total biomass, might have greater internal stem damage. They just accumulate this through time or as they grew. And we thought that species with denser wood would be harder for the termites and the microbes to decompose. So denser wood would lead to lower internal stem damage. So what did we actually find? Well, here you can see that we have our precipitation gradient with our 5 sites. So these are our 2 rainforest sites. These are our 2 savanna sites. And here you can see the amount of termite damage on down deadwood. So there was definitely greater termite activity, greater termite damage at our driest sites. If we then go and look at our measure of internal stem damage, we see that our driest sites had the highest total amount of stem damage. There was more stem that was missing or had somehow been compromised at the driest sites than at the wettest sites. And you can see we move from savanna to rainforest. And this is true for all 3 of these graphs. The x-axis moves from left to right, increasing moisture content, or savanna the left 2 and rainforest in the right 2. The other thing we found, which was pretty surprising, was that the diameter didn't change. The average diameter was not significant across our gradient. We didn't get necessarily bigger trees in the rainforest, which is what we expected. And finally, the densest wood was at the driest sites, which wasn't really surprising. Many studies have shown that you get increasing wood density at drier sites. When we looked at these together, what we found was that the internal stem damage was best predicted by the site level variables, both precipitation and the amount of termite damage in down deadwood. So you get greater internal stem damage at the driest sites, as well as where there's greatest amounts of termite damage in down deadwood. What was not expected and what was pretty interesting was that their interaction, that both of them interacted with wood density. So basically what we found was that the lighter wood, the things that had less dense wood, had the most internal stem damage at the rainforest sites, which is probably an indicator of fungal heart rot or microbial heart rot. The thing that shocked us was that we actually had the greatest internal stem damage at the densest wooded species in the savanna. And thinking about it afterwards, we've got this Coptotermes that likes to build mounds at the base of trees and then hollows the inside of trees and lives inside them. So all of us want to live in a strong and sturdy house. So they're taking the densest wooded trees that are typically in the eucalyptus, the myrtaceae, and hollowing the insides of those to live inside them. And so we had this really surprising interaction with wood density and our site level variables. And when we wanted to take this and scale up to say, what does this mean at the landscape? Well, if you look at green is the total amount of above ground biomass we estimated at each of the sites. We see that the rainforest and the sclerophyll site has the greatest total amount of above ground biomass than the 2 savanna sites. But then if we look at how much of that above ground biomass was compromised, we see that that the amount of damage was almost 37% in the driest savanna and 22% in the wetter savanna. So a quarter to a third of our trees were either missing or damaged in the savanna, but only 6 to 7% were missing in the rainforest. So there's a lot of differences across our gradient. There's a comparable measure in Borneo where in lowland rainforest, they had about 8% of the tree was damaged. in the Bornean rainforest. So our rainforests are pretty similar to other places, but nobody's measured this, quantified this in savanna. And so if you're trying to account for carbon and think about how much biomass is there, then you're going to do a really poor job, especially in savanna systems. This also made us realize that we're losing a lot of our biomass. I've always thought about measuring the down deadwood, and that's where you start looking at biomass loss from wood. But in our system here in the savannas, a lot of that biomass is actually being lost while the tree is still alive. And so we've started understanding our system in far north Queensland and starting to build these ecosystem scale models. But this told us only about what's happening in these far north Queensland systems. And I really wanted to expand out to think of what was happening at a more global scale. And so for that, we scaled up. And how we did this was that when we were setting up the pilot study, we were in James Cook University and talking to Mirko Karen, who is part of the Turn Ecosystem Network, and or Supersite Network, sorry. And so he was saying that they were doing lots of things. They had flex towers and plots, and they were measuring all this productivity, but they weren't doing things like looking at how fast the carbon was leaving these forests. And so he really liked our simple protocol and suggested that they put it out across Turn. Alex and I happened to be up in Borneo talking to Kate Parr and Paul Eggleton, who work in various countries in Africa, as well as Borneo in Asia. And so they agreed to put out wood blocks there. I happened to be on sabbatical at the time in Bordeaux and started talking to different friends and collaborators as I gave different talks or went to different conferences, and so convinced people that they wanted to join in the effort too. And so we really went global. And then as I started and managed to talk to Juan and convince Juan that he should put out sites in Colombia there. So that's how we ended up with amazing sites that I got to visit in January. And then as we look for holes in the data or holes in the locations, I went onto social media, so Twitter and Facebook, and recruited even more people in that way. And so it was really interesting to see all the stories and ways that people came into the project. We set up a Google Drive repository for people that have protocols and post their data and things like that. And so if you go onto Twitter, you can see here's our site in the Alps going in Italy, where Alberto has convinced his dad to help carry blocks up into the Alps to deploy our project. And here's our site going in New Zealand. And so we ended up with 108 collaborators at 133 sites in 20 countries and 6 continents. And so here, let's look at the story itself. And so we were able to publish this in Science back in September. We made the cover with these are wood-feeding termites, the Microceratermes from our rainforests in Australia. So pretty fun to make the cover. Perhaps even more fun was that First Dog in the Moon, which is a satirical column, and The Guardian in Australia picked us up and got the story. They picked up the story very well with the small caveat about how they represented rainfall, but you can't complain too much when First Dog in the Moon covers you. And so one of the things the First Dog in the Moon posed was whether termite scientists, if there's a better job in the world. And First Dog in the Moon said no, and I would agree. It's been really fun to explore the world through the lens of termites. And so in this project, what we asked was the relative climate sensitivities of wood-dwelling microbes and wood-feeding termites. Our expectation was that microbial decay would be highest in warm, wet locations, and termite discovery and decay would be highest in warm locations, but less sensitive to precipitation. So this really follows in what I gave you in that initial primer of thinking about the different sensitivities that they might have. And so here you can see the distribution of our sites around the globe. And so we certainly have large gaps in places we weren't able to get sites deployed, but we did a really nice job, I think, in deploying across the latitudinal gradient. So we're equally represented in the northern and southern hemisphere, and we were equally represented in the tropics and the temperate zone. And so what everybody did was they sourced locally radiata pine, with a few cases not radiata pine, but for the most part radiata pine. They encased that radiata pine in mesh that excluded termites and then cut holes in half of the bag so that we could allow or exclude termites. And you can see here we have Whitaker's biomes. So we have mean annual temperature and mean annual precipitation. We didn't get any sites in the tundra and only a few sites in the boreal forest and the temperate grasslands and deserts, but otherwise we have a pretty good representation across different biomes. This is the line for the tropics and subtropics here. Here's an example of wood that was harvested after a year where at the same site termites found it or termites didn't find it. And this is our block from the pilot that for me had the highest termite decay. So when I put it out, it weighed 180 grams. When I harvested it, it weighed 300 grams, but only 3 grams of that was wood that was still remaining. So massive amounts of soil import into this bag by the time I harvested it at the end of a year. To explore the climate sensitivities, we looked at decay as we use the decay constant K, which is a negative log of final mass divided by initial mass divided by time. And we separated out if the termites discovered the wood or if the termites didn't discover the wood. And because I think these are two very different trajectories if the termites have actually gotten in and started eating the wood. And then we looked at, of the termite treatment, what percent of termite included wood actually had evidence of termites? What percent, when they were allowed to discover, did they actually discover? Then we, for each of these response variables, we looked at mean annual temperature, mean annual precipitation, and their interaction term. Okay, so what did we find? Well, here's our first. Here you can see microbial decay. Microbial decay was only sensitive to temperature. Interestingly, it was not sensitive to precipitation or the interaction. Here you can see decay, the decay constant. Higher decay constants mean faster decay rates. And then you have mean annual temperature on the x-axis. You can see that we had a Q10 of 1.7. This was not significantly different to 2. So we recovered what we found in smaller scale studies, that you have essentially a doubling of microbial decomposition with a 10-fold increase in temperature. Let's shift to termites. Again, when you allow termites into the wood and they discover it, so you have termites plus microbial decay, they're only sensitive to temperature. And again, precipitation was, and its interaction was not a significant predictor. So again, have increasing decomposition, increasing K values, and mean annual temperature. What you see is that the Q10 in this case was 6.9, almost 7. So you had, I think, a 3.7-fold increase in, when termites discover the wood, a 3.7-fold increase in your Q10 value with increasing temperature. So they're super, super responsive to temperature increases. We don't know why, but we have some ideas and we're thinking about exploring these in various ways. So I'd be happy to chat with anybody as this is interesting to you. We were so shocked by this Q10 that we kept rerunning it to make sure we didn't make mistakes. Finally, if we look at termite discovery, what we see is that finally we have a variable that's sensitive to temperature, precipitation, and their interaction term. So you have higher discovery on the y-axis. Again, you have mean annual temperature on the x-axis, and then we have precipitation represented by this bar. So the wettest sites are in blue, and the driest sites are in yellow. If we first start by looking at the wettest sites, what we see is that you have a positive relationship that as you increase in temperature, you have greater discovery. What was really interesting and to some degree surprised us was the degree of sensitivity discovery had at the driest sites. So at the driest sites, as you increase temperature, you have much more discovery at the driest sites. And so overall, then you had the greatest termite discovery at the driest versus the wettest sites in the warmest places in the world. You can see this, so I didn't tell you before that all of these dots, the color coding was discovery. So the darker the color means a higher discovery rate. And so you really can see that tropics effect with this line. So you have the tropical rainforest, the tropical seasonal forests and savannas, and you have the subtropical deserts. And you can see that the highest discovery, these darkest, that discovery really increases as you move into the tropics and subtropics, and especially at these driest sites. Taken together, you can put the pieces together, both the discovery and decay. So here you have decay constant K, and you go from boreal forest all the way up to the subtropic and tropical sites with the subtropical deserts, the tropical seasonal forests and savannas, and the tropical rainforests. The box plot on the left is where you have the undiscovered wood that's just undergoing microbial decay. And on the right, you have the discovered wood that has microbial plus termites. You can see that termites always elevated the decay rates, but they elevated them the most at these warm, dry sites of the world. Really interesting. So we were understanding much more about how termites and microbes were affected by these different climatic gradients, how sensitive they were. But we wanted to take this one step further. We wanted to think whether we could use our climate sensitivity to predict areas with high discovery into the future. And so what we did was we took our discovery model and we said that we're going to say that high discovery rates are if it's greater than 50%. And we combined this with WorldClim downscalings of the CMIP6 climate models. So we took the stronger model and the weaker model. Our stronger model said that things were going to get a whole lot worse than they are now. The weaker model said that things were actually going to get better than they are now. And we modeled this into mid-century. So what we found was looking at by mid-century with our stronger and our weaker model. We have in gray is continuing where currently there's almost no termite discovery, and we don't expect that that's really going to increase in the future. In that bright green, we have we expect it to continue to be low discovery by mid-century, so greater than 5%, but less than 50%. In that pea green is where we already have really high discovery at greater than 50%. In the buff color is where we're unable to predict that amount of discovery. And then the really interesting one for us was this orangey gold color. And this is where currently there's low discovery, but by mid-century, we expect that we're going to increase to high discovery rates. And you can see that in both the stronger and weaker model, you get upticks in where we expect high termite discovery, mainly in the Southern Hemisphere, especially in Africa and in Latin America, but also in places in Australia. So we're expecting that with warming temperatures, with tropicalization, we're going to increase where termites can discover the world, which means that we expect that the rates and forms that carbon is going to cycle in these places will also change. And so you can see this here. Here we have the global mean terrestrial warming by mid-century relative to a baseline between 1970 and 2000. And then the y-axis is increasing the terrestrial area with high termite discovery. So you can see across both models, this strong positive increase as you go from 1 to 5 degrees temperature warming compared to baseline, the amount of area that's discovered. So overall, microbial decay was highest in warm locations in the world, but interestingly not wet locations. The termite discovery and decay was highest in warm locations, but only discovery was sensitive to precipitation. And then finally, we predict this large expansion of high termite discovery by mid-century with tropicalization. And there's some caveats to that I'd be happy to chat with you further about if you're interested. But we think that this can lead to really big differences in how carbon is being handled in these landscapes. Okay, so that was our story, which was really quite an adventure to carry out. And I want to tell you a little bit about the authors. And so for me, I find it's really important to think about doing collaborative, inclusive, and open science. So what does that mean? Well, for collaborative inclusive, we came up with an accessible protocol. We had no centralized funding for this project. All the PIs had to come on board with their own resources. And so that meant that I had that grad student in Brazil who was carrying out this project with local resources. And I also had PIs, such as Juan, who were able to put in multiple sites at their field site and involve different people in their lab. And so I was able to get buy-in at a lot of different scales of researchers. It was also bottom up. So we took something that was a very simple protocol, which meant we could do it in a lot of places. And then we could ask a big question with this, again, with no centralized funding. So it's a pretty different model to our typical funding models. When I apply to NSF with big budgets and I say that I'm going to do high delivery, I don't often deliver a science paper with this. So it was pretty fun to see how we could populate up to big scales with a bunch of small pieces. I worked really hard at recruiting. Certainly I could have recruited more places and there are gaps in the landscape, and I've since been contacted by people who want to add into some of those places. But I worked really hard to try to access people at lots of different scales. And I've never done anything like this with recruiting on places on social media to get projects. And some of the conversations that led to, this was done across COVID lockdown. And so I was having these amazing conversations of people's realized lives across that as they were trying to put out the experiment or trying to join in. It was really important to me to make sure that everybody who contributed to make this project possible was co-authors. And so there was some concern about this along the way where one of the big distributed sites, people thought that they were putting things out, but weren't going to be recognized. So I made sure that people knew that they, once I realized that everybody was going to be included on this paper. And so it was inclusive with co-authorship, and then worked hard at recognition. So I think the day before we released, I looked up every single name of our 180 names to see who had a Twitter profile I could follow so I could tag them when I posted our story. One of the things that was super interesting, because it was COVID, I hadn't met many of these people or hadn't seen them for quite some time. So many of the co-authors are people I don't know, never mind them meeting one another. So I had a Zoom that I held 2 Zoom calls 12 hours apart the day that the paper was released. And so in this call, we have all 6 continents represented. We had 2 people from Africa and only 1 person from North America. So did a pretty good job of making it so that people around the globe could hop online to meet one another and exchange their stories. What were their experiences they deployed this experiment? I also think it's really important to have open science. So this was required by science anyway, but it's something that aligns with what I think is really important. Is one, it's good so that people can check to make sure that what you say you do. So you're able to put your code and your data and make it available to the world. So we have a GitHub repo for our code, and we also posted everything up in Figshare. So we have the data, and this is a growing database. I think it's also interesting because I can think of the ways I want to use my data. But I can't, but every time I've released different products into the world, people have come up with really creative ways to make use of my data I never dreamed about. And so it's also led to some really interesting collaborations or novel uses of data. Given that most of what we do is funded by our taxpayers, I think it's nice to be able to open this up to lots of different uses of the data we produce. And so it was really nice to do this. And so you can go online and you can explore the data that we collected and see what we did. Finally, I want to take a couple minutes to just talk about the illustrator of these amazing pictures that I've shown you across today. So as I was first in Cairns, Australia, staying near the Botanic Garden, I was setting up the study with the big, I just got my NSF NERC award, and I went to the Botanic Garden to look at a basket weaving exhibit in the Tanks Botanic Gardens art galleries. And as I was there, I heard somebody around the corner saying words like mycorrhizae, decomposition, microbes, trees, and pulled me into this other room. And I came across Donna Davis and her exhibit. She was with a mycologist too. And she had had a fellowship where she worked with the Botanic Garden in Queensland and collaborated with botanists and mycologists. And she spent a year going out each week to this endangered Melaleuca forest and looking at all the fungi that she found that were the fruiting bodies that were there. So she did things like took spore prints and screened them onto silk so that people could see in big, bold ways what spores looked like. She also represented the tree and fungal diversity by having different amounts of jars. And she had various things you could touch and look at and move, and things like this box that was suggesting what was going to happen to diversity in the face of global change. So I found this a really compelling exhibit, and I ended up taking my funding from my NSF project for broader impacts and giving it to Donna so that we could have a collaboration together. You can go to her website and read more about our project. She came out to the field with us twice, and so asked lots of questions that pushed us to really think about our system as she developed projects. You can follow her on Twitter and Instagram, see some of her amazing exhibits. Here's Steve Allison, my collaborator, draped in one of her pieces where she screened these termites onto material. Because in the exhibit I saw that everything was very tactile, but because we did this internationally and across COVID, a lot of what she developed with us were videos. So if you go to Vimeo, you can view a bunch of her videos where you can see kaleidoscoping termite mandibles tied to music that somebody wrote as we described what we thought termites sounded like when they chomped on wood. Here you can see this is a gauge, an old school gauge for measuring changes in temperature, and you can watch as this material decomposes. She's had a number of exhibits across Australia and also in Miami. And I've done 3 science artist discussions with her, the panel discussions, which have been fascinating as we both talk about our creative process. It's amazing how similar that creative process is for scientists and artists as we develop our work. You can go to the Lowe Museum in Miami, at University of Miami, has a virtual exhibit, so you can actually tour around in our exhibit. You can read essays, you can watch our discussion panel. This is one of my favorite works she's done, and so you can see these kaleidoscoping termites, wood, and microbes. Each of these frames represents those taxa in the system. So these are actually photos that my grad student Becca took of termites, and it represents our gradient. So this can be seen as a graph. So you can go from low termite diversity to high termite diversity across our gradient here, kaleidoscoping around. And what Donna has really done is push me to think about science communication. So people learn in lots of different ways. Some of those can be reading papers or listening to a scientific talk like today, but others are amazing visuals or watching videos that Steve Allison made of our termite explorations. And I've done a podcast with the release of the science paper to reach out to different audiences and explaining. So I think everybody in my group really benefited by working with Donna, and everybody's gone on to think of different ways to display visually or through audio aspects about their science. And Donna was really a glue across COVID, bringing our labs together in virtual space to do things like coming up with a termite game and things like that. So with that, I'm going to stop and take any questions. I'm going to leave you looking at this. Here's a series of fun facts that Becca wrote and that Donna put to artwork. And so thank you so much for your attention and listening to me today. And I'd be very happy to take any questions.
Thank you so much, Dr. Amy. We are going to start the question sessions because we just have until 2 p.m. So you can raise your hand on or write your question or comment in the chat, and please in English. Entonces, por favor, si alguno tiene una pregunta o alguno tiene una pregunta, levantemos directamente la mano o bien la pueden escribir por el chat, por favor. Ok, Profesor Posada, gracias.
Thanks, Amy, for, do you hear me? Yeah.
Yep.
Thanks, Amy, that was great. Very nice to see this extended presentation. I really enjoyed it. I had a question about another study you presented before, and was what links did you find between wood traits and fire, other than bark, I guess, because bark size is a key trait, but did you look at other traits like wood anatomy, density, and what did you find?
Yeah, great question. So we did not directly study fire. When I was putting in the NSF grant and to ARC and also to NSF, we kept getting the fire people who pushed back and said, well, you're not going to have anything left after fire, so why would you study how traits relate to fire? Or we had people say, well, that's not the right amount of fire and things like that. So in the end, we didn't include fire as one of our manipulations, although we planned it initially. But of course, in our driest sites, these are fire-prone landscapes. So we had 2 fires go through at our very driest site. And so I don't think that we had wood burn. We did as best we could to put fire breaks in and things like that. So I don't think it was a proper study to really look at how wood traits related to fire. And so far, we've really just focused mainly on the biotic decayers. And then I've started looking at solar radiation in other studies because things like lignin are photoreactive pigments. If you think about, like, if you wear your blue t-shirt, your favorite blue t-shirt out, blue, that'll be photoreactive and it'll bleach through time. Lignin is like that too, and you'll get bleaching and releasing CO2. So we've done some work on photodegradation in the lab, but not properly studied fire. So great question. I do not have the answer, sadly, for you.
Okay. Interesting to look at.
Mm-hmm.
Yeah. Thanks.
Yes, Nicola.
Yeah, thanks a lot for the very nice presentation. And I was curious, just a very short question. What's next? What follows with termites? What do you, if you can say it?
Oh, sure. I'd be happy to. Yeah, great question. A bunch of different things. So one of them that we've really been pondering with the termites is why they're so reactive. And it could be something about populations, you know, that they can just have more babies over more periods of time when you have warmer periods for longer periods of time. So it's a population number thing. Another could be that we've been looking at the decay enzymes, the cellulases themselves, and that they have an optimal temperature, surprisingly around 40 degrees Celsius. So it could be that they don't work that well and then all of a sudden start working really well at warmer temperatures. And so we actually have to get into the gut physiology and trying to understand how these enzymes work. We found that there's in Antarctica, there is a cellulase that actually has a lower optimal temperature. So there is evolution of different optimal temperature among the microbes that do this decomposition. So I think it might be a really weird, I have to get into insect gut physiology, and finding somebody who does that has been a little bit challenging so far. So if you know of anybody, do let me know. So there's that aspect. I'm also really interested in following up on those termites here in Brazil that build the mounds and thinking about how they affect the CO2 and methane efflux at the ecosystem scale. So we're starting, I've got a grad student who's Brazilian who's starting to work in our savanna systems here and the flooding system. So playing around with that a bit more. I think I have another answer for you, and if I remember it, I can email it to you, but I'm not remembering off the top of my head. A bunch of different termite studies.
Lots of interesting questions.
Another that I would like to do, if anybody's interested in, is a global poop project. So thinking about how efficient termites and what comes out of termites on the other end, because they can't break down lignin. And so what are they actually defecating and how are they using that to build their mounds? And so thinking that we can collect from different animals eating vegetation and measure how efficient they are at processing plants.
Wow.
The other.
Good luck. Lots of interesting questions and challenging.
Yeah.
¿Alguno más? Somebody else? I don't think so.
And you can feel free to pose in Spanish, and I'm sure somebody will help
Okay.
translate. I'll understand half of it.
It would also. No. Let me see in the chat. Okay. No. Okay, Dr. Amy, thank you so much for your time and for joining us with your interesting research. Muchísimas gracias a todos por participar y a todas por participar el día de hoy. Próximamente cargaremos el video en YouTube y nuevamente se da el espacio para agradecerles y que nos sigan acompañando en las diferentes sesiones del seminario. Muy buenas tardes y un cordial saludo. Gracias.
Hi, Amy. Thank you very much.
Thank you so much. And if anybody has questions, feel free to directly email me. Juan can give you the email if you don't have it. So good to see everybody. Hope to talk to you soon. Bye,
Thanks
Nicola. Bye,
again.
Juan. Thanks, Veronica.
Bye, Amy.
Ciao, ciao.
Ciao.
Thank you very much.
Chao.